Welcome to Science Era. Today's topic is immune system. The immune system is a network of molecules, cells, tissues and organs, each with specialized role in defending the host against extracellular microorganisms, cancer cells and intracellular parasites. While we discuss innate defenses and adaptive immunity separately for clarity, the recognition and removal of damaged host cells and foreign substances is an interconnected and highly regulated symphony of events connecting both arms of the immune systems. We're going to discuss the cells and tissues that respond to mediator and to the damaged cells and invading substances. So, starting from the cells of the immune system, let's begin. So the three main objectives of today's lesson will be recognize the different type of leukocytes involved with innate resistance, outline the leukocyte response to microbial invasion, and then integrate leukocyte distribution with host resistance. Starting with the cells of immune system. We're going to name the cells, the main cells of immune system first, and then we're going to look at each one in detail separately. So the first cell is called granulocyte. Then we have mast cells, monocytes and macrophages, dendritic cells, lymphocytes, each has a specialized role in defending host. And then we have leukocytes. Are known as white blood cells, which are involved in both specific and non-specific immunity. All arise from the pluripotent stem cell. The figure shows the different type of human blood cells. Pluripotent stem cells in bone marrow divides to form two blood cell lineages. So the first one is called myeloid progenitor. The common myeloid progenitor cell give rise to the erythroblast that produces erythrocytes or red blood cells, megakaryocytes that produce platelets, and unknown precursors that give rise to mast cells. The granulocytes, three types of granulocytes are there: eosinophil, basophil, and neutrophils. Then monocytes that give rise to dendritic cells and macrophages. The second type of lineage is lymphoid progenitor. Lymphoid progenitor cells give rise to natural killer cell as separate lineage of DC or dendritic cells. The B cells that become antibody secreting plasma cells and the T regulatory helper and cytotoxic cells. Note that dendritic cells can arise directly from myeloid and lymphoid progenitor cells and from the non-proliferating monocytes. Starting with mast cells. Mast cells specialize tissue cells similar to basophils that trigger local inflammatory reaction and are responsible for many allergic symptoms. They are bone marrow derived cells that differentiate in connective tissue. Mast cells have an indented nucleus and a cytoplasm filled with specialized organelles called granules. Mast cells are non-phagocytic, but when stimulated, they rapidly release the content of their granules into extracellular environment, a process called degranulation. Mast cell granules contain histamine, po- prostaglandin, serotonin, heparin, dopamine, platelet activating factor, and leukotrienes. Because these compounds influence the tone and diameter of blood vessels, they are termed as vasoactive mediator. So the examples of all the vasoactive mediators are histamine, prostaglandin, serotonin, heparin, dopamine, platelet activating factors, and leukotrienes. Additionally, mast cells possess high affinity receptors for the types of antibody associated with allergic response. When they become coated with this type of antibody. Binding of antigen to the antibody triggers the release of performed vasoactive mediators. These vasoactive mediators plays a important role in allergic responses such as eczema, hay fever and asthma. So in order to summarize mast cell, we say that they are specialized tissue cells similar to basophil that trigger local inflammatory reaction and are also responsible for many allergic symptoms. Next one we have is granulocytes. Granulocytes are of three types: eosinophils, basophil, and neutrophils. Granulocytes have irregularly shaped nuclei with 2 to 5 lobes. Their cytoplasm has granules that contain reactive substances that kill microorganisms and enhance inflammation. So, as we said, three types of granulocytes are eosinophil, basophil, and neutrophil. Eosinophils are active in worm and fungal infection, allergy, and inflammatory reaction. Basophil function in inflammatory events and allergies, while neutrophil are essential blood phagocytes active engulfers and killers of the bacteria. Basophil. Here you can see the diagram on the screen of a basophil. Basophil have irregularly shaped nuclei with two lobes. The granules contain histamines and other substances similar to those in mast cells. However, basophil arise from different cellular lineage. Like mast cells, through basophil tend to infiltrate specific tissue site rather than circulate through the bloodstreams. Basophils are also important in development of allergies and hypersensitivities. Next one is eosinophil. Eosinophil also have two-lobed nucleus connected by a slender thread of chromatin. Eosinophil granules contain hydrolytic enzymes, nuclease, glucuronidases and peroxidases in addition to caustic protein called major basic protein. Unlike basophil, eosinophil circulates in low numbers and migrate from bloodstreams into tissue space, especially mucous membranes, when recruited by soluble chemotactic mediators. They are important in defense against protozoan and helminths parasites, mainly by releasing enzyme, cationic peptide, and reactive oxygen species into extracellular fluids. These molecules damage parasites' plasma membranes, killing it. Eosinophils also play a role in allergic reactions as they have granules that contain histaminase and aryl sulfate. These are the down regulators of inflammatory mediator histamine and leukotrienes respectively. Thus their number circulating in bloodstreams often increase during allergic reactions, especially type 1 hypersensitivities. Last type of granulocyte is neutrophils. Neutrophils are highly phagocytic cell with a nucleus that has 3 to 5 lobes connected by slender threads of chromatin. Because of irregularly shaped nuclei, neutrophils are also called polymorph nuclear neutrophils or PMNs. Neutrophils have conspicuous organelles known as primary and secondary granules. Primary granules contain peroxidase, lysosomes, defensins, and various hydrolytic enzymes, whereas smaller secondary granules have collagens, lactoferrins, catechisins, and lysozymes. These enzymes and other molecules help digest foreign material after it phagocytosed. Mature neutrophils leave bone marrow and circulate in blood so they can rapidly migrate to a site of tissue damage and infection, where they become a principal phagocytic and microbicidal responders. Neutrophils also use oxygen dependent and oxygen independent pathways that generate additional antimicrobial substances to kill ingested microorganisms. Let's summarize what we learned about granulocytes. So there are three types of granulocytes: neutrophil, eosinophils, and basophils. Neutrophils are also known as polymorphonuclear nucleophilic leukocytes or PMNs, also called polys. Their morphology include lobed nucleus, granules in cytoplasm, and amoeboid appearance. Location in body accounts for most of the circulating leukocytes, few in tissue except during inflammation and in reserve nucleus. Reserve locations. The function of neutrophils is phagocytize and digest engulfed materials. Then second type of granulocyte is eosinophil. They are large eosinophilic granules, non-segmented or bilobed nucleus. Few in tissue except in certain types of inflammation and allergies. They participate in inflammatory reaction and immunity to some parasites. Next one is basophil, or which is about 0 to 1% in the blood. They have lobed nucleus, large basophilic granules, basophils in circulation, mast cell present in most tissues. Function is the release of histamine and other inflammation causing chemicals from the granules. Monocytes. Monocytes are mononuclear leukocytes having granules in the cytoplasm and an oval or kidney-shaped nucleus. They are created in bone marrow and enters the bloodstream where they grow and migrate into tissues. Mature into macrophages or dendritic cells after roughly 8 hours of circulation. Macrophages are organelles found in mononuclear phagocytic leukocytes that are necessary for phagocytosis and contain a plasma membrane covered in microvilli. They bridge innate resistance and adaptive immunity. They have PRRs that detect certain PAMPs, and phagocytosis is induced when their PRRs binds to them. They can also move to lymphoid tissue as activated cell to deliver antigens to lymphocyte. Macrophages appears to excite T and B cells that have already been sensitized to antigens. Macrophages are immune cells that spread throughout the body and settle in specific organ where they are referred to be fixed macrophages. Moving on to dendrite or dendritic cells. They are a heterogeneous group of cells with neuron-like appendages. They arise from various hematopoietic cell lineage. Both lymphoid and myeloid dendritic cells are known. They present in small numbers in blood, skin and mucous membrane of nose, lungs and intestines. They are programmed to detect and phagocytose foreign substances, especially pathogens. Also express pattern recognition receptors (PRRs), contact phagocytose and process antigen, then display foreign antigens on their surface. This is called antigen presentation. Let's summarize mononuclear phagocytes of three types: monocyte, macrophages, and dendritic cells. Monocyte, they have single nucleus, abundant cytoplasm. In circulation, they differentiate into either macrophages or dendritic cells when they migrate into tissue. Function is to phagocytize and digest engulfed material. Then coming next coming on to macrophages. They have single nucleus, abundant cytoplasm. They present in virtually all tissues, given various names based on the tissue in which they are found. Phagocytize and digest engulfed material. Same function as monocytes. Then we have dendritic cells. They are branched initially in tissues, but they migrate to lymph nodes and other secondary lymphoid organs. They gather antigens from tissues and then present it to lymphocytes that congregate in the secondary lymphoid organs. This brings us to the lymphocyte. Lymphocytes are the major cells of immune system. Major population include T cells, B cells, and natural killer cells. B and T lymphocytes differentiate in bone marrow from stem cells. They are only activated by binding of specific antigen onto lymphocyte surface receptor. After activation, replication continues as lymphocyte circulates and enter lymphoid tissue. The memory cells are activated lymphocytes that do not immediately replicate but will do so later in host's life when antigen is again present. So the three types of lymphocytes are B lymphocyte, T lymphocytes, and natural killer cells. B lymphocytes. B lymphocytes mature in blood marrow. They circulate in blood and can settle in lymphoid organs. After maturation and activation, they are called plasma cells and produce antibodies. Activated B cells also produce memory cells that are primed to act upon subsequent exposure to antigens. Then we have T lymphocyte cells. They mature in thymus. They can remain in thymus, then circulate in blood or reside in lymphoid tissue. Like B cells, requires antigen binding to surface receptor for activation and continuation of replication. Activated T cell differentiate into two types: helper T cell or Th and cytotoxic lymphocyte CTLs. They secrete cytokines. Chemicals that have effect on other cells are produced and secreted by activated T cells. This figure provides the summary of the development and function of B and T lymphocytes. B cells and T cells arise from the same lineage but diverge into two different functional types. Immature B cells and T cells are indistinguishable by histological staining. However, they express different proteins on their surface that can be detected by immunohistochemistry. Additionally, the final secreted product of mature B and T cells can be used to identify the type of cell. Memory B cell initiate rapid response to reinfection with same agent. Antibodies neutralize toxins and virus opsonize bacteria. Then in T cells, Th cells growth and differentiate factor enhances or suppresses the immune cell's action, and CTLs, lytic enzymes and proteins, kill altered or infected cells. Next we have is natural killer cells. Natural killer cells are a small population of large non-phagocytic granular lymphocytes that plays an important role in innate immunity. The major NK cells function is to attack and destroy malignant cells and cells infected with microorganisms such as viruses and intracellular bacteria. However, NK cells do not recognize antigens. They recognize their targets in one of two ways. So, two ways of recognizing target cells are: first, bind to antibodies which coat infected or malignant cells. which is known as antibody dependent cell mediated cytotoxicity, ADCC. Then recognize cells that have lost their class 1 major histocompatibility agent antigen due to presence of virus or cancer. The figure on the screen represents the system used by natural killer cells to recognize normal and abnormal cells. The figure A on the left is normal cell express a ubiquitous membrane protein known as the class 1 MHC. Cells that are infected or becoming cancerous may lose the class 1 MHC protein and express stress or shock protein. Figure on the right side is the NK cell evaluates each cell it contracts for the presence of class 1 MHC or stress protein. If no class 1 MHC is found and or stress proteins are found on the cell, the NK cell use cytotoxic granules containing perforin and granzymes to induce cell's death by apoptosis. This figure shows the antibody-dependent cell-mediated cytotoxicity. In Figure A, in this mechanism, antibodies bind to a target cell infected with virus. In B, NK cells have specific antibody receptors on their surface. In C, the antibody bridges the infected cell with NK cells so that the target is close enough for the enzymatic attack. Let's summarize the blood stem cells. So, blood stem cells are divided into two groups, myeloid stem cells and lymphoid stem cells. Lymphoid stem cells have lymphoblasts, which are of two types, B lymphocytes, T lymphocytes, and natural killer cells. On the other On the other hand, we have myeloid stem cells, which contain red blood cells, platelets, and myeloblasts, which give us granulocytes, which are of three types: eosinophil, neutrophil, and basophils. Eosinophils, neutrophil, basophils, B lymphocytes, T lymphocyte, and natural killer cells all combine to form white blood cells. This was all about the cells of immune system. In the next lesson, we're going to discuss organs and tissues of the immune systems. Thank you for watching. If you like my video, don't forget to subscribe for more.